I will share with you is this, that our data suggest that each oil has its own biochemical fingerprint. A fingerprint that we believe underlies the biological activity of an oil, and importantly, we think can be utilized to assess the quality of oils, even to the point of detecting adulterated or synthetically built oils. And we'll show you that from a biochemical standpoint for the first time today. So, before I put my science hat on, if I can just briefly introduce my institution and our research team. Roseman University is a private, not-for-profit health sciences institution. We offer accredited doctoral and postdoctoral programs in pharmacy, dentistry, and orthodontics. We offer accredited undergraduate nursing programs along with an MBA. We currently have 1,600 students enrolled from 43 states across the U.S. and 16 countries throughout the world. We're really proud of our students and the programs that we offer. I'll tell you that Roseman has a strong teaching mission, but also a research focus. I know that we're not in class. I typically don't have 10,000 students when I do take the opportunity to teach, but just as a quick aside, we are a mastery learning institution, 90% pass-fail, so please make sure you're taking notes. We'll give a quiz here in a little while. So that's our institution. Here's us, our research team. Very proud of what we do in the College of Pharmacy. We have a group of interdisciplinary scientists who bring a tremendous breadth of expertise from pharmacokinetics, medicinal chemistry, biomedical engineering, biochemistry, and pharmacology, all with the primary research interest in developing nutraceuticals as therapeutic agents. And importantly, we are interested in perhaps changing the narrative, in increasing the understanding of how nutraceuticals affect health and well-being, and to increase the understanding, particularly among medical and scientific communities. So here we are, that's what we do, and I'll highlight Dr. Tim Lee and his postdoctoral research associate, Dr. Yasuo Arasaki, who much of the work that I'll be presenting today has been done by them. Now, one last indulgence for 30 seconds and just one slide. Our research team in just the last few years have had their research efforts recognized by news and media outlets, both nationally and internationally. These are just a few examples, including Newsweek, the Houston Chronicle, LA Times, Chicago Tribune, yes, the Deseret News here locally, the Times of India, the Independent in the UK. What I haven't listed here is another 150 news and media outlets across Europe and Asia. We really do have an excellent group of scientists that have been gathered and been very productive. So, some of you may remember a syndicated radio host from years back named Paul Harvey, and he had a program called The Rest of the Story. Anyone remember The Rest of the Story? Everyone that just clapped is over the age of 40. I think is how that works. The rest of the story was this. Mr. Harvey would tell little known details to give the story behind the story. And today I feel it's particularly important to do just that, as Dr. Hill alluded to. The rest of the story for our relationship with doTERRA began in a barn about 2 years ago. Now it's a pretty barn, a picturesque farm that's near Mount Timpanogos. At a social event that was being held there, I had the opportunity to interact with someone who I later learned was a doTERRA representative. Once I realized that, I was very eager to bring our conversation that day back to science. I wanted to tell him that our research team had independently been working on doTERRA oils to study them. And I thought doTERRA, Dr. Hill, would be very interested in some of our findings. Now let me just share with you some of the things that we did with, unbeknownst to doTERRA and in complete independence, we went to the website and purchased a variety of essential oils. And then we went to competitors, even major competitors, and purchased their analogous oils to study. Now, some of the projects that we've had going on, we're very interested in the pharmacokinetic properties of essential oils. This essentially is, how are those oils absorbed? How are they distributed throughout the body? Where do they go? And how are they metabolized? That's one project. Earlier today, Dr. Hill shared with you another project of ours where we've been interested in the effect of essential oils on biomarkers of oral health. He shared with you, I believe, the activity of an enzyme called MMP8, matrix metalloprotease. It's an enzyme that breaks down healthy gum tissue. The lower the enzymatic activity, the healthier the gums. One of our scientists discovered that On Guard is a potent inhibitor of MMP8. Now, yes, that's right. Those are interesting findings. The last project I'm going to tell you about, as a prelude to today's discussion, is the antioxidant properties of doTERRA oils. Now, I know that for all of you, that is not a surprise to anyone in this room. And in fact, it wasn't a surprise to us. We purchased ylang-ylang and black pepper and ginger, and analyze them with scientifically accepted methods for determining antioxidant activity. As expected, we found that each of them possessed robust antioxidant properties. Here's where the surprise was to us. When we deconstructed the oil and purified the major constituents, each of those major constituents on its own could not reproduce the antioxidant effect. That's right. And I'll allude to what that means here in a moment. More importantly, perhaps, we tried to synthetically reconstruct the oil, putting back in the major constituents. That synthetically built oil also did not possess antioxidant properties. Right. Now, the last little tidbit I'll share from those early studies is this, and you may or may not be pleased to hear this again. I'm sharing this as an objective scientist. The essential oils that we purchased from doTERRA competitors lacked significant antioxidant activity. And we found that to be curious. So here you go. We've always got our thinking caps on. These repeated observations began to suggest something that we hypothesized and we call internally now in our group the oil effect. Let me tell you a little bit about what we think our observations imply for an oil effect. If that's true, these are the observations that we have made. One, that doTERRA oils have robust, therapeutically relevant signaling effects on human cells that we can measure through scientifically validated research models. Two, that those effects are induced by the whole oil, not the individual components of the oil. And then three, as that last statement suggests, at least in our hands and with the oils that we had, we did not see comparable effects with doTERRA competitors. So that barnyard conversation that happened a few years back, this was the essence of it. And again, we suggested that perhaps these independent findings that doTERRA would be interested in, as Dr. Hill alluded to, he's a very smart man. And we had some extended interaction with him. But in the meantime, we still were interested in what we thought was a very curious and potentially important discovery about the oil effect with respect to doTERRA oils. So with that in mind, let me just describe to you briefly the process of scientific discovery. Essentially, as scientists, what we do is we spend a lot of time trying to prove ourselves wrong, which if you ask my wife, she'll say, for me, that's not very tough. But we work hard, we make a discovery, and immediately turn around and try to poke holes in that discovery and determine if it will stand up to further scrutiny. And that is exactly what we did. Everything that I'm about to share with you moving forward is the work of Dr. Tim Lee and his postdoc, which I referred to earlier, Dr. Yasuo Hirosaki. Now, we pulled out the big hitters. If you're here tomorrow and listen to Dr. Lee, you'll see that he is not necessarily my size, but he is a great scientist and has done some excellent work. What we did is utilize some advanced methodology and instrumentation to study the cellular effects of essential oils. What we wanted to do was determine whether our oil effect hypothesis would stand up to significantly increased rigor in testing. So I'll describe to you some of Tim's methodology here in a minute, Dr. Lee. But just to tell you that that was the initial purpose of these studies. But Dr. Lee realized as he was planning those experiments that one byproduct of these studies is that we would identify a biochemical fingerprint for every essential oil that we tested. Now, what does Dr. Lee do? I'm going to introduce you the concept of nanofluidic proteomics. Proteomics is the large-scale study of proteins. Proteins throughout every cell in the body. Now, simply put, proteins are the basis of life. Proteins and their activity. The body's ability in cell systems, organ systems, the ability to respond to environmental changes for us, to live day to day, relies on our ability to regulate those proteins and enzymes. So Dr. Lee has been utilizing this nanofluidic proteomic technology to study other disease states like diabetes, fatty liver disease, more recently cancer biology. And what we did was brought essential oils to that table and utilized this methodology and Dr. Lee's expertise to do the same thing, to study the cellular effects of essential oils. So, how does it work? If you want to know, all you have to do is read that slide. Nanofluidic proteomics via high throughput and multiplex capillary isoelectric focusing amino acids. So, there's no reason for you to remember that. Well, actually, there's one. The next time you're at a party and you need to get out of a conversation, just drop proteomic profiling via capillary electrophoresis. After the dazed look goes away, people remarkably have somewhere else to be. Not that I know that from personal experience, it's just hypothetical, of course. So the picture of that instrument there, this nanofluidic proteomic system is a robotic system. It's automated. We like that because robots don't sleep and don't get tired. It allows us to get a lot of information over a short period of time. In very basic terms, what happens is that system will take all of the proteins within a cell and separate them. It pushes them into a microcapillary tube and separates them by charge and by size. And then Dr. Lee can take very specific antibodies and look for changes in proteins that regulate their activity. Now, these are called post-translational modifications. And you don't need to remember that either. But I want you to remember one type of post-translational modification, and it's called phosphorylation. Now, here's what these experiments look like. So we performed, or Dr. Lee performed these studies in human hepatocytes. So these are liver cells. Everything that I'm about to share with you of his work was done in human liver cells. Culture them in dishes, and then treated those cells with essential oils for 24 hours. This is not a transient effect. This is after a long-term treatment. And then utilizing nanofluidic proteomics, Dr. Lee was able to probe multiple signaling pathways. Now let me briefly describe these pathways. There are 3 listed on the screen there. Again, I know this looks complicated. The one on the left is called the MAP kinase pathway. The one in the middle is PI3 kinase, and the one on the right is JAK-STAT. There are many other signaling pathways. These are pathways that control how your cells both behave and respond. Now let me make 2 points. One is that you'll notice that in each pathway there are multiple proteins associated with it. In that sense, it's like a row of dominoes. Once you push one over, then it will activate a cascade that leads to, and this is the second point, a larger both cellular response and even a systems biology response. So for example, the MAP kinase pathway, when it's activated, is associated with cell growth and rejuvenation. The PI3 kinase pathway, when it's activated, involves and influences metabolism. And the JAK-STAT pathway is involved with immunity or healthy immune responses. Now, last point on this slide, and then we'll get going with some data. And this is very important to remember, that each of these pathways needs to be able to be accelerated or slowed down, depending on the cellular context. It's very important to be able to turn these systems up or down, depending on what is needed physiologically. With that in mind, let's look at what this looks like. So Dr. Lee first used Copaiba, the doTERRA Copaiba, which is an important note. And this is 24-hour treatment of human liver cells with Copaiba. What you're seeing here is a chromatogram. The top level, the top graph, is cells that have not been treated. That's just what normally happens. And the bottom graph or curve is in response to the treatment with Copaiba. I'll try and make this as painless as possible. Just very quickly, if you look on the right, those are unphosphorylated or inactive enzymes. In this case, we're looking at MEK1. So in resting cells, there's a lot of inactive MEK1 enzyme. After treatment with Copaiba, the amount of inactive, unphosphorylated enzyme is decreased. The amount of activated or phosphorylated MEK1 increases significantly. That helps us to know that there's an increase in phospho-MEK, or the activated formation of MEK1. Dr. Lee did similar studies using MEK2. And again, you'll see that after 24 hours of treatment with Copaiba, there were increases in the phosphorylated or activated forms of MEK2. And finally, ERK1 and 2, you'll see the same scenario. Ultimately, this leads us to conclude that Copaiba in human liver cells activates multiple components of the MAP kinase cascade. As far as we're aware, using this instrumentation, these are the first studies of any kind of this type in nutraceutical development and essential oils. But that's just one pathway. What about the others? And now that you know how to read a chromatogram, I'm going to go through this rather quickly. So this is the AKT phosphorylation, part of that PI3 kinase pathway. Untreated cells, there's lots of phosphorylated or activated form of AKT. After 24 hours of treatment with Copaiba, you can see a significant, in this case, decrease of phospho-AKT. Another protein called 4-EBP1, which is downstream of AKT, you can see also an inhibition of phosphorylation of that enzyme. Am I speaking Greek to anyone out there? Yeah. I know. It's suffice it to say that through these studies, we now know that Copaiba inhibits PI3 kinase pathway. Lastly, but not least, STAT3 Copaiba increases dramatically the phosphorylation or activation state of STAT3. Here we go, now we're getting to the good stuff. So in a summary, Copaiba, we now know, has a dramatic and varied effect on cell signaling pathways in human liver cells. We repeated those studies, or Dr. Lee did in his laboratory, with green mandarin, helichrysum, melissa, pink pepper, and turmeric. Look at that list. What you are seeing, first of all, for the first time, this has not been shown elsewhere that we are aware of, anywhere, is that these doTERRA essential oils have an exquisite ability to fine-tune up or down regulation of signaling pathways that change physiology. What you're looking at is the biochemical fingerprint of each of those oils across these pathways. It's a remarkable achievement of Dr. Lee and his laboratory.
Wow.
But now we get to the good stuff in these last few minutes. So if you've been asleep, now's a good time to wake up. Now that we know that we can generate that biochemical fingerprint, what can we do with that ability? Well, what we did was take some of those competitor oils and test them against the cellular effects of doTERRA oils. We took 3 competitors, so we're looking at green mandarin, turmeric, and pink peppercorn. We used this proteomic profiling across those 3 pathways. Let me show you what are some very interesting results. So, and I promise, I think you'll find that this is worth it. That first graph at the very top are the effects of green mandarin on JAK-STAT signaling. You can see that in untreated cells, there's very little phosphorylation. In cells treated with green mandarin, there's increased phosphorylation, which means that green mandarin activates that enzyme. When we, under the same conditions at the same concentration, took a competitor oil in that assay, we found not just no effect. We found that the competitor oil inhibited the JAK-STAT pathway. Now, Dr. Lee is a very smart scientist, and he wondered, he had a suspicion, if the effect of that competitor oil was the result of a single aromatic constituent. In other words, was this a spiked oil? And so he just simply purchased some limonene from a lab supply company and put it into those cells. Do you see any similarities? What Dr. Lee discovered, we think for the first time, is that using that biochemical fingerprint gives us the opportunity to screen for adulterated oils.
Wow.
And I'll come back to this point in a minute, and at least from our perspective, again, an independent, unbiased perspective, we believe that that's a significant point. That it's possible to try and disguise chemistry, but your cells do not lie.
Right.
It is not possible to disguise biology or to fool biology. Now again, from trying to have a reasonable and sound scientific approach, this could be a one-off, so we simply did it again with a different oil. Now we're looking at turmeric versus a competitor. And again, you can see after turmeric treatment with human liver cells after 24 hours, there's robust increases in the phosphorylated form of, in this case, it's MEK1. That means that that protein is being activated. The competitor, again, inhibited the activation of MEK1. So again, we were interested in whether that could be the result of a spike, perhaps. So just simple administration of aromatic turmerone produced the same effect as the competitor oil, which again in our hands suggests that perhaps it was an adulterated oil based on the biochemistry.
Shame, shame, shame.
And then lastly, looking at the PI3 kinase pathway using pink pepper, where the doTERRA oil decreased activation of AKT, we saw with a competitor there was no change, and with spike with alpha-pinene, we saw similarly no change. Now, just these last few moments, here's a summary slide. I know that looks like a lot of color, but look at what it tells us. That the green mandarin doTERRA effect on those pathways was dramatically different than a competitor.
Fantastic.
What wasn't different was the effect of the competitor oil versus buying some very cheap lab supply oil. They looked the same. That was true for turmeric and pink pepper. Now, just a couple of conclusion slides. If we were in class, at the point I say that, usually there's an applause. But let me, oh yeah, I wasn't asking for one. We can save it. I'm glad you're not that eager for me to finish. 2 slides to summarize. First, the major findings from Dr. Lee's work, we believe, confirmed our hypothesis about the oil effect concerning doTERRA oils. there's a robust physiological, quantifiable effect in human liver cells that have the potential to affect many different therapeutically relevant pathways. The effects of the oil are with the whole oil, not the components. And as I said earlier, competitor oils either had no change or inhibitory effects, and that those effects were mimicked by single constituents. This is a significant slide. For me, this next one really hits home. What are the implications of the oil effect? If indeed that's true, what it tells us is that the quality of essential oils is the combination of chemistry and biochemistry. I'll say that differently. Biochemical analytics, we believe, are required to fully assess the quality of essential oils.
Yes.
As we were talking the other evening, it occurred to me, and I hadn't ever before thought of it this way, in this sense, this biochemical fingerprint has become, for us, a sure lie detector test
Yes.
for adulterated oils from a physiological standpoint, which means that the therapeutic potential of oils is highly dependent on the complete composition of the oil, and the natural conclusion of that is sourcing, processing, and characterization of the oil, from our perspective, is everything, from a biological perspective.
Great job, doTERRA. Gold standards.
And lastly, we just feel like that opens up a lot of opportunities for qualitative assessment and, to a greater extent, quantitative assessment of these effects. Now, you just survived a lot of science in a short period of time. I hope that you can take away some key messages, one of which is in our hands, and again, I'm here as an independent scientist, in our hands and in our assays, doTERRA oils, from a biochemical, biological standpoint, do seem to be superior. And we feel like with these pathways, we've been able to define and explain perhaps why that is, at least from that perspective.
Phenomenal.
Lastly, I hope that you will come tomorrow for Dr. Lee's talk. If you think anything that I've presented today of his work is surprising, he has some data tomorrow that he will share, which from my perspective is stunning, where he went to great lengths to exhaustively compare doTERRA oils using these assays against a major competitor. And the results were surprising even to us from an objective standpoint.
Fantastic.
Thank you for your time. If you wouldn't mind, I think my call